Hi, very good afternoon to everyone. At the outset, I would like to thank the organizers, Simulation Plus, for organizing such an interesting workshop and inviting me to share my views. Today, I'm going to talk about the application of PBBM modeling in generic product development in terms of regulatory applications. And I will also talk about few of the case studies. Moving on to the next slide. Uh, a disclaimer, the opinion expressed here are solely those of mine, and they do not represent the statements or opinions of Dr. Reddy's Laboratories Limited. So in the talk in the next half an hour of this presentation, I will be talking about what PBBM modeling is primarily in the area of generic product development, in terms of the PBBM model development, general modeling workflows, dissolution data integration, biopredictive versus QC dissolution media, case studies. I'll be touch base on the couple of case studies, primarily focusing on the biowaiver of lower strength, where we see a F2 mismatch, and also biowaiver for a BE study, which was leveraged from one market to another. And the third case study I'm going to talk about is about the dissolution specification justifications, or relaxation of the dissolution specifications for a modified release product. I'll be talking about some of the regulatory deficiencies which we have been getting on the PBBM the modeling part, and finally on the conclusion. Moving on to the next slide. So this slide basically talks about the role of PBBM modeling in the drug product life cycle. Although we have applications of this in preclinical, as early as preclinical discovery and even phase 1 to phase 2, but from a generic medicine perspective, the application starts as early as phase 3, phase 4, and continues till phase 4 post launch of the innovator drug, and further launch of the generic medicine. In terms of the application, the areas where we could use PBBM modeling, it could be used for setting up a BE safe space. It could be used about for widening the in vitro dissolution specification, which I'm going to talk about in for as one of the case study. It could be used for setting the patient-centric dissolution acceptance criteria. It could also be used for getting waiver of the Fed studies as well. So, and also one important application of PBBM is to identify the CPP, CMAs, and CQAs, which can ultimately impact the CBAs, that is the critical bioavailability attribute. And more importantly, this approach can be used to address the deficiencies or queries from a biopharmaceutics perspective from different regulatory agencies as well. Moving on to the next slide. This is more specific to the application of PBBM in the generic space. We know that PBBM is a tool which combines API, formulation, pharmacokinetics with physiology to enable the prediction of the in vivo exposures in both fasting as well as fed conditions. And as we see of the diagram on the right hand side, you see that it has numerous application, starting from extrapolation from preclinical to clinical, for predicting the drug-drug interactions, for food effect predictions, for the confidence on the B assessment between the reference and the generic, for getting the biowaiver for the lower strengths, for dissolution safe space. Again, I will talk about this example in the subsequent slide. For development of clinically relevant dissolution specifications, F2 mismatch, and even in setting up the particle size distribution specifications as well. So this approach, as we see, it has numerous applications, and these approaches are more and more being accepted by different regulatory agencies, including FDA, EMA, as well as other agencies. Moving on to the next slide, this kind of gives us the overview that how different regulatory agencies are have come up with different guidance documents on the reporting structure or analysis of data using the PBPK model, and also for the format of the reports. FDA is very open. Even other agencies are open to discussions in this area, including the IMA. Even WHO has a guidance way back in 2010 for characterization and application of PBPK models for the risk assessment. Even apart from these agencies, other agencies in our experience, the Envisa, the MedSpace, the Chinese regulators are also, as well as the Japanese regulators as well, are open to these kind of novel approaches. And this screenshot also gives a summary of the workshop which was jointly held between different regulatory agencies. Moving on to the next slide. So this slide for the PBBM model development, it gives a general workflow. It primarily involves three main steps. First, the model setup, then the model validation, and finally the model application. In terms of the model setup, we will look into the biopharmaceutics properties, the formulation aspects, and the physiology. This kind of information we can get from the literature. We can generate experimental data, or we can also use tools like the ADMET predictor to arrive at those parameters. However, it is important that we have adequate justification for the all the input parameters which we have used during the model setup. The next phase comes in terms of the model validation, where we could use internal validation as well as external validation. And typically, we use the same criteria of prediction error, what we use for the IVIVC. In this, the validation can be done against the literature data. It can also be done against in-house data. And basis this, we can perform population bioequivalence as well as virtual simulations using the representative populations which have been used in the clinical study. Once this is done, the setup, the validation part, then comes the application part, where we could derive conclusions from a physiological perspective, where we have numerous applications where we could use it to establish the to get the waiver for the lower strengths. We could also use to justify any formulation changes, manufacturing changes, as well as to get waiver for the food effect studies as well. Moving on to the next slide. Again, this slide kind of talks about the dissolution data integration, which is a very important aspect from a modeling perspective. We have divided this into, this is again taken from our recent publication which came just the last month in APS Pharmaceutical Science Technology. We have divided the incorporation of the dissolution data basis the different dosage forms, like immediate release dosage forms, modified release formulations, and delayed release formulations. In terms of the immediate release dosage forms, we can use the particle size input from the API PSD. We can use the Johnson model. If model is not sensitive, we could also use the dissolution input as direct. And alternatively, we could also use the PPSD approach, which I'm going to talk in a little bit more detail in the subsequent slide. We could use the Z factor. We can use single Z factor, or we could use the multiple Z factor, and we could also generate the Z factor versus pH profiles, and we can validate other batches. With using these approaches, we can get support for the dissolution specification setup, setting up of the safe space, and also the clinical impact of the CBAs. For the modified release formulations, we could use again the direct dissolution input, or use the most commonly the Weibull fitting. We could fit single Weibull, double or triple Weibull, and could also establish IVIVC or IVIVR. The third case is the delayed release dosage forms, where we could use the Z factor or also use the direct dissolution input. In case of the in disease factor, we could also use for products which act locally in the intestine, products which act in the colonic region, and we can set up the conditions accordingly to suit the formulation type. Again, this has also applications for setting up dissolution specific support or the generation of the dissolution safe space. Moving on to the next slide. Again, as I said, we are going to talk about the PPSD model, which can be used using the triple D plus module. We can develop the PPSD model to determine the API PSD in the formulation. And this figure describes that. Just take an example of a formulation A, where we can fit in the dissolution profile by varying certain parameters. Once we fit the dissolution profile at a particular pH, we can get the optimized PPSD. And as you can say, in this case, we have obtained the mean PSD. This PPSD can be transferred to GastroPlus without the need of additional dissolution input. However, you also have to validate this for the formulation under different pH conditions. Like here, we have validated for pH 4.5 and pH 6.8. Similarly, for formulation B, where you can see the release profile is slower, and then the mean PSD which is coming out is on the coarser side. But basically, what we are trying to say is this is an alternate approach which can be done, but PPSD needs to be further validated with additional media and formulation variants. Moving on to the next slide. Again, this is a very important concept about the QC media versus biopredictive dissolution media. See, if currently all regulatory agencies are based or justifications which we make primarily are based on the QC media. However, it may not be biopredictive. If the QC media is also biopredictive, then we can consider the molecule or the product on which we are working as very low risk, because this is the best case, and we can have the same method which can be both used as a QC release as well as biopredictive. As we know, QC is routinely used, and it should have the ability, that method, dissolution method should have the ability to reject the non-BE batches based on this dissolution media. But most of the time, this is not the biopredictive. Hence, there is a need to develop a media, a separate media, which is biopredictive, and a method that can predict quantitatively the in vivo outcome and help develop the IVAVR and a PBBM model. However, if we don't have a biopredictive media, then we will have challenge in answering a lot of regulatory questions and need to rely on the biopharmaceutics risk assessment. And then the product or from modeling perspective, it comes under the high risk category. So in nutshell, what we are trying to say is it is important to develop a QC media which should also be biopredictive. And now moving on to the next slide. See, this is another very important concept of the dissolution safe space. In this graph, if you see on the left side, we've taken the innovator drugs, and on the right side, we have taken the generic drug. So this is the generation of data for the pivotal test and RLD. And other batches where which we have tested in the BE studies. If we have a data which we can differentiate between the non-BE lots and the BE lots, then we can very well define the BE safe space. As you can see on the right graph, the batches which are bioequivalent and close to the pivotal test and the RLD, they can be considered to have the let me just use the pointer. Yeah. So if you see here, this is the region where the pivotal test and RLD is there, and the other batches which have been tested and found to be bioequivalent. Whereas if you see on the extreme two lower in vitro release profiles, these are the batches which were not bioequivalent, and hence basis this we can establish the safe space, and overall we can consider this as our knowledge base. Moving on to the next slide. So now we have talked in general about the modeling, how to develop the model. Now I will walk you through some of the case studies which we have successfully been either submitted or they are under review with different regulatory agencies. The first case study which I'm going to talk about is for a biowaiver of a lower strength of a product where there was a F2 mismatch. So this was a weakly acidic BCS class 3 compound which had 5 strengths for ranging from 40 to 320. We did the BE study at 320 milligram. This compound has a solubility low solubility under acidic conditions, and the solubility increases in basic conditions. So as I said, we did a fasting bioequivalence study at 320 milligram. And in order to obtain the biowaiver for the lower strength, especially as per the EMA guidance, we have to generate the comparative dissolution at 3 pH. In all the pH conditions, the requirement of F2 greater than 50 was met. However, for 40 milligram at a specific pH, which is pH 4.5, the F2 mismatch was there, and we were getting a F2 of 43. As you can see in the right graph, that the test product on the lower the 320 milligram pivotal test is in blue line, and the below is the test product. And since we could not show the similarity between this, the agency, we were asked by the agency to conduct the BE study at lower strength to justify the approval for the 40 milligram strength. So here we have used the PBBM modeling approach to support the biowaiver for 40 milligram study, where we had seen the faster dissolution profile for the one of the strength, and we compared it with the BE strength using the pH versus Z factor approach, as we will see the details in the subsequent slide. Moving on to the next slide. As you can see in this slide, we followed the traditional approach here, which is the model development. We validated the model using the pivotal B data in fasting conditions, used virtual B simulation, and then for model application, we used the dissolution profile of the 40 milligram into the 320 milligram model, and proved that there was no impact under in vivo conditions in spite of having a difference between the lower strength and the higher strength. And there was no impact of the failing BE study on the biowaiver for the 40 milligram. So hence, basis this approach, the biowaiver was granted. If you move to the next slide, this kind of gives the approach here. If you see, we use the 40 milligram dissolution as the input, and we showed bioequivalence with the 320 milligram test product as well as the 320 milligram reference product. And as you can see the numbers here, both the T by R ratios as well as the 95 confidence, 90% confidence interval were within the 80 to 125 range. And basis this, we were able to justify the biowaiver for the 40 milligram, and we got the approval for this, and we could prove that there is no impact of the F2 on bioequivalence, and biowaiver for 40 milligram was granted. So we can see from this example that using this approach, if we do proper validation and have sound scientific justifications, we could save both cost, time, and as well as it could lead to the early launch of the product. Now, moving on to the second case study. The second case study is a case where we had done the BE study for market A, and we got the product approved. And then we were leveraging the same BE study for market B. Whereas in market B, when we were supposed to compare, we were leveraging the BE study, but we were supposed to compare the in vitro dissolution of the test product with the market B reference product. When we did that comparison at a particular pH, especially pH 6.8, we found that the F2 was not matching for both 100 milligram as well as the 50 milligram strength. As you can see in this slide, the first, this shows is 1.2 pH, 4.5, and then below is the is 6.8. This is for 100 milligram, and the right panel is for 50 milligram. As you can see in the lower panel, that at pH 6.8, we were not able to show the similarity at pH 6.8 between the other market reference product as well as the pivotal test product. Hence, we were asked to justify this. We were either we had to repeat the BE study with the reference product of market B, or we had to justify using a modeling approach. So we use the same approach to justify or support the waiver for the BE study for both 50 and 100 milligram. Moving on to the next slide. As you can see, I'm giving an example of the higher strength. The same is applicable to 50 milligram. Here, what we used is we used the we validated the model. We can see that the values are pretty much comparable between the observed and the predicted values in used obtained in the pivotal study for market A. And then we compared the pivotal reference used for market A versus market B reference. And we also showed where the concern was coming, that market B versus the pivotal test, we could show that in spite of the dissolution differences in pH 6.8 and F2 not matching, we could show based on the virtual bioequivalence after thorough validation of the model, that it has no in vivo impact. And as you can see, the T by R ratios as well as the 90% confidence interval is within the specified range. And hence, we received favorable feedback from from that particular regulatory agency, and we could avoid doing the BE study, which again led to a huge saving of cost, time, and also early market entry. Now moving on to the so now we have talked about the primarily the BCS1 BCS class 3 compounds, where we could use PBBM modeling for biopharmaceutics risk assessment. And this chart or this decision tree is something which is nowadays being used or and also recommended by the regulatory agencies to be followed. And where in cases where we have permeability limited absorption and we could develop the PBBM model, there is a possibility to get waivers or and get in more number of candidates for the bio waivers, as well as to once we establish the dissolution safe space, we can get extension of the dissolution specifications as well as flexibility in the manufacturing site, if we can establish the dissolution safe space as well as the PBBM modeling approach for the products which have permeability limited absorption. This is commonly used for biopharmaceutics risk assessment. Now moving on to the last case study. So we have talked of two cases where we were leveraging or the BE study, and we were trying to get biowaiver for the lower strength. This is the case where this was a extended release formulation which had 5-point specifications, but based on the stability data, we wanted to relax the existing specifications from 50 to 70%, which is traditional plus minus 10%, to 45 to 70%. And the proposal was made specifically focused on 7 hour to relax from 50 to 70 to 45 to 70%. And again, in this case, we used or we took help of the PBBM modeling to justify this dissolution specification through virtual BE and IVIVR. Moving on to the next slide. As you can see, we first validated the model. We validated for the reference for the pivotal test product, and we could explain the population. We could capture the variability obtained in the study. Once we did the validation both with the in-house data as well as with literature data, we kind of generated the lowest low profile dissolution profile, as well as a single point lower dissolution profile, where we changed the 7-hour time point to the lowest spec which we wanted to have. And using this dissolution profile as input, and after thorough validation and doing virtual bioequivalence, we could show that we could establish the bioequivalence between the reference product, as well as using the single point low dissolution profile for the point which we wanted to have a relaxation. And as we can see, we got the ratios well within 80 to 125, as well as the 90% confidence interval for this dissolution profile, as well as the extreme case of the dissolution profile where we had taken all the lowest point were within the bioequivalence limit. Basis this, we could get a favorable outcome for this approach to get the relaxation for the dissolution specifications. So with all this, we have given a kind of a glimpse that how PBBM can be used for different applications. And in these cases, we had used extensively GastroPlus, Triple D Plus, and other tools to arrive at these justifications. Now, moving on to the next slide, to give you a glimpse of some of the recent queries which we have been getting from the regulatory agencies in terms of the PBBM justifications. The some of the queries we get is how to justify the model inputs, whether they are based on experimental data or literature data. We have to have a parameter sensitivity analysis to justify selection. Then there are also questions around the Z factor for dissolution input. What is the basis or scientific rationale for selection of the Z factor and demonstrating the relation of Z factor to the absorption dissolution curves? Then other factors include as I've talked about, the relevancy of the QC media and its ability to reject non-BE batches, where we said we could develop biopredictive medias. There were also questions around the mechanistic framework of the model or of understanding the ADME process. And then there are also comments on the consideration of the CBA, that is the CPP CMAs, which are used in the model. So we have to include the product quality attributes while building the model. Obviously, validation is one of the most important aspect of the PBBM modeling, and validation against pilot BE batches, especially failed BE batches. Then there are also questions around the discriminatory power of the QC media. And then in recent times, we are also getting certain queries on the incorporation of the female volunteers in the healthy volunteer BE studies, because in countries like India, due to cultural reasons, it is difficult to enroll equal proportion of females in the BE studies. So we have also used this approach to kind of justify, along with the literature support, to assess the impact of the gender on the pharmacokinetics of the drug. Finally, coming to the conclusions, we have talked about that PBBM modeling has demonstrated applications in both generic and innovator domain. And apart from regulatory agencies like FDA, other agencies like Anvisa, MedSpace, Chinese regulators are also open on such modeling justifications. And we have given certain case studies where we could avoid the BE studies in case of we could relax dissolution specifications, and also could justify the F2 mismatch to get the biowaiver for the lower strengths. The current focus of PBBM modeling could be on the biopredictive ability of the QC media, certain regulatory justifications to understand the mechanistic framework, and ability of the biopredictive media to predict the failed BE batches. And some of the upcoming areas of research could include the justifications for waiver of FED studies, and multiple dose studies may be focused on BCS class 1, class 3 kind of molecules. Overall, as we see that this approach is getting a lot of attention, and more regulatory agencies are open to such submissions. But the key point is knowledge sharing amongst different regulatory agencies, academia, as well as industry is a must. Now, in the end, I would like to acknowledge Shiv Charan from Biopharmaceutics team, the entire Biopharmaceutics team at the Global Clinical Management Group at Dr. Reddy's. I would like to thank Dr. Reddy's management for providing all the infrastructure and support, all my cross-functional teams, collaborators, and regulators who have contributed to the data generation and feedback for these case studies. And last but not the least, simulation team for giving me this opportunity and organizing this wonderful, very interesting workshop and thought-provoking workshop to bring all the people together. These are some of the references, some of our published articles which we have referred in the presentation, and we have been publishing extensively in this area, and we are still learning and trying to develop in this area. Finally, I would like to thank all of you. Thank you very much for your patient learnings, for your patience and bearing with me. If there are any questions, I'm more than happy to answer. Thank you.